Welcome back to Science Impact. Today, we're diving into a fascinating paper about tackling a really nasty environmental pollutant, cadmium.
Oh, cadmium. I know it's bad, but how bad is it really? And where do we even encounter it?
It's pretty serious, actually. Cadmium has no beneficial role in our bodies, and it's highly toxic. It comes from industrial uses like batteries and leaks into our food and water.
So it just hangs around, like it doesn't break down easily in the environment or in us?
Exactly. Once it's in us, it accumulates in organs like kidneys and liver for decades. This causes oxidative stress, DNA damage, and can lead to serious health issues.
Wow, that's quite alarming. What about current solutions? Are there ways to get rid of it?
Existing chelation therapies, like EDTA, have limited success against tissue bound cadmium. They can also have side effects, which isn't ideal.
So we need something better, something safer and more effective. Is that what this paper is all about?
Precisely, the researchers developed something called a short metallopeptide conjugate, or SMPC. It's a biomimetic nanostructure designed for both detecting and detoxifying cadmium.
A dual function solution? That sounds incredibly clever factorial. How does this SMPC actually work though?
It's quite ingenious. The SMPC uses precise metal chelation, meaning it grabs onto cadmium ions. This binding also triggers a stimuli induced self assembly process.
Self-assembly, you say? Like, it changes its shape or structure when it finds cadmium?
Yes, exactly. It undergoes a morphological conversion. It starts by forming tiny cadmium nanoparticles, which then transform into less toxic nanorods.
So, it doesn't just bind it, it actually changes the cadmium into a less harmful form? That's amazing.
And it does this while also enhancing fluorescence, which allows for real time detection and quantification of cadmium levels. So you can see it working.
That's a fantastic combination. Detection and detoxification in one go. What's the SMPC made of technically?
It's a short peptide, specifically a pyridine bis tyrosine conjugate. Its crescent shape is key to selectively binding cadmium ions over other metals like iron.
And they confirm all this with experiments, right? Like, how did they know it was actually binding and changing form? Uh,
They used a whole suite of techniques UV, IR, and fluorescence spectroscopy confirmed the strong binding. NMR and FTIR showed the molecular interactions happening.
okay, so lots of lab tests to prove the chemistry. But what about seeing the actual transformation from nanoparticles to nanorods?
That's where transmission electron microscopy, or TEM, came in. They could literally see the spherical SMPC structures nucleating cadmium nanoparticles, then evolving into elongated nanorods over time.
Visual proof factorial. That's always so compelling. Did they test if it actually worked in living systems too?
Absolutely. They started with human embryonic kidney cells, HEK293 cells. Cadmium alone significantly reduced cell viability, but with SMPC, viability was largely restored.
So it protected the cells from cadmium damage. That's a huge step. What about a whole organism?
They used zebrafish, a common model organism. Cadmium exposure caused severe developmental abnormalities like malformations and reduced survival.
And the SMPC helped the zebrafish too, I hope?
It did. When SMPC was introduced, it substantially alleviated these developmental defects. The peptide itself was also shown to be biocompatible and non-toxic to the fish.
That's fantastic news. So it's not just a lab curiosity, it has real potential for protecting living things.
Indeed, this work shows SMPC can clear cadmium and reduce oxidative stress, which is major mechanism of cadmium toxicity.
This really seems like a game changer for heavy metal detoxification. Imagine this being used in environmental cleanup or even in medicine.
It's a truly transformative strategy. This human and environment friendly platform opens up new avenues for biomaterials in monitoring and detoxification.
It's exciting to think about the future applications. A simple, biocompatible solution to such a persistent and dangerous problem. What a brilliant piece of research factorial.